Hello everyone. In this video, we are going to discuss the TCA cycle in detail. TCA cycle are tricarboxylic acid cycle. We know that in glycolysis, glucose is converted into pyruvate. Glucose gets converted into pyruvate. Next, this pyruvate is converted into acetyl CoA. This pyruvate gets converted into acetyl CoA, and then this acetyl CoA is oxidized via TCA cycle, and energy is released, which is used by our metabolic system. So how this happens basically, the first step is transfer of this pyruvate from cytosol into the mitochondria as TCA cycle. TCA cycle occurs in mitochondria. The pyruvate which is formed in cytosol is transferred into mitochondria, and it happens by specific pyruvate transporters. Once pyruvate is inside the mitochondria, it is converted into acetyl CoA, and this reaction is catalyzed by PDH complex or pyruvate dehydrogenase complex. PDH complex is a complex of different enzymes with different catalytic activities, which catalyze the conversion of pyruvate to acetyl CoA. Pyruvate is a 3-carbon compound, and acetyl CoA is a 2-carbon compound. So basically, one carbon is released in this process in the form of carbon dioxide. Does this PDH complex comprises? It has basically 5 enzymes: E1, E2, E3, and one is kinase domain, PDH kinase, and one is PDH phosphatase. So it is basically a complex of 5 enzymes which perform their respective catalytic activity. E1 is regarded as pyruvate decarboxylase, while E2 is regarded as dihydrolipoyl transacetylase, while E3 is regarded as dihydrolipoyl dehydrogenase. So these are the three catalytic activities, and kinase and phosphatase, these play a role in the regulation of this complex. How this enzyme system works, we should know before the working the cofactors or coenzymes of these different domains. So basically, this E1 has coenzyme TPP or thiamine pyrophosphate. E2 has coenzyme lipoic acid and CoA, and E3 has coenzyme NAD and FAD. So basically, this enzyme complex has 5 coenzymes. So how it works? This is pyruvate CH3, C double bond oxygen, C double O negative. So this is the pyruvate, and what happens in the first step? Thiamine pyrophosphate, which is the coenzyme of E1, combines with it, and in this step, the carbon dioxide is released from the pyruvate. So as carbon dioxide is released, its space is occupied by thiamine pyrophosphate or TPP, and a intermediate hydroxyethinyl derivative or intermediate is formed. And this reaction which is happening here is basically catalyzed by pyruvate decarboxylase or E1. So this derivative or intermediate is formed, and this intermediate in turn combines with lipoic acid. This is the chemical formula of lipoic acid, but for the sake of convenience, we write this domain as L, and two sulfur are regarded as S. So we write a shortened formula of lipoic acid for our convenience, because we have to deal only with sulfurs here, not deal with carbon, hydrogen, or oxygen. So what happens? This intermediate combines with lipoic acid. Okay, and what happens next? Again, one more reaction occurs, and in this reaction, one more intermediate is formed, which is CH3, C double bond oxygen, SLSH, and the thiamine pyrophosphate, which was used as coenzyme and was bound with this intermediate, which is released here or recycled here. So this one intermediate is formed, and this reaction is catalyzed by E2 domain of PDH complex, and it is dihydrolipoyl transacetylase. Now this intermediate combines with CoA. It combines with CoA and gets converted into acetyl CoA, CH3, C double bond oxygen as CoA. So this is the acetyl CoA or required product, and the lipoic acid is reduced into LSH as such 2 hydrogen are donated to lipoic acid in this process. So in order to regenerate lipoic acid, E3, the third domain of PDH complex, works, and its coenzyme is FAD. This FAD reacts with this reduced lipoic acid and gets converted into FADH2. And in this reaction, original lipoic acid is regenerated. So what happened? This FADH2 in turns reacts with the second coenzyme, which is NAD. It reacts with NAD and it's get converted into NADH plus H, and original FAD is regenerated in this reaction, and this reaction is third is catalyzed by dihydrolipoyl dihydrolipoyl dehydrogenase or E3. So let's have a brief overview what is happening here. Basically, first of all, this was the pyruvate, and this pyruvate reacted with the thiamine pyrophosphate or the coenzyme of E1, and this reaction was catalyzed by E1 domain of PDH complex, and this reaction carbon dioxide was released and thiamine pyrophosphate get bound to it and form an intermediate. Next, lipoic acid, which has chemical formula this one, but for the sake of convenience we replace C8H14O2 with L and wrote is in the simplified form, and this lipoic acid reacted with this intermediate and a new intermediate was formed, which is written here, and this reaction was catalyzed by the second enzyme or E2, and then CoA came, the third coenzyme of this PDH complex, and it reacted with this intermediate and gets converted into acetyl CoA or required product of the reaction. Next, in this step, the lipoic acid was reduced and by donation of 2 hydrogen atom, and then FAD comes and takes 2 hydrogen from this lipoic acid, and original lipoic acid is regenerated and the FAD is converted into FADH2, and next NAD reacts with this FADH2 and gets converted into NADH plus H, and this reaction is catalyzed by E3. So basically three domains of PDH complex played their role and acetyl CoA was produced. So next we come toward the regulation of this complex. First thing you should keep in mind that TCA cycle does not have hormonal regulation. It does not have any kind of hormonal regulation. So it is regulated only by its products, different allosteric regulators, activators, inhibitors, but it does not have any hormonal regulation. So PDH complex is basically regulated by its two enzymes which are part of it, which is kinase and phosphatase. So what happens when kinase is activated is phosphorylates. When kinase is activated, it phosphorylates E1 domain and inhibits it. Upon kinase activity, E1 is inhibited and this PDH complex working is inhibited. So when this kinase activity is stimulated, this kinase activity is basically stimulated in presence of high energy signal. So what is high energy signal? High energy signal is basically ATP and NADH. So when there is surplus ATP or NADH in cell, it stimulates the kinase activity of PDH complex, and kinase activity phosphorylates E1 and inhibits it, saying that there is no need of further ATP or NADH. So this reaction stops. So there is no formation of acetyl CoA. Furthermore, it is also stimulated. It is also stimulated by acetyl CoA. So when there is sufficient acetyl CoA, it signals that acetyl CoA is surplus. So there is no need of making further acetyl CoA. Again, kinase activity is stimulated and E1 is phosphorylated and inhibited. So this is the stimulation of kinase activity and inhibition of E1. When kinase is inhibited by higher concentration of pyruvate. When there is accumulation of pyruvate in the cell, it signals that pyruvate is too much and this pyruvate needs to be converted into acetyl CoA, and this pyruvate inhibits kinase. As a result, E1 is stimulated and acetyl CoA is formed, and this reaction proceeds in forward direction. So this is the regulation of E1 and kinase activity. So how phosphatase is regulated? Phosphatase is basically regulated in response to calcium. Calcium is the activator of phosphatase activity. So when there is sufficient calcium inside the cell, it stimulates phosphatase activity, and the E1 gets dephosphorylated, and again it becomes stimulated to convert pyruvate into acetyl CoA. And when it happens when muscles are contracting skeletal muscles have higher concentration of calcium in cytosol, and this higher concentration of calcium in cytosol stimulates phosphatase activity, which dephosphorylates E1 and activates it. So this is the basically regulation of PDH complex. It does not have any kind of hormonal regulation. This kinase and phosphatase activity are cyclic AMP independent. They does not depend upon cyclic AMP or glucagon or insulin for their activity. They are working independently of hormonal regulation. So what happens in case of PDH deficiency? The pyruvate dehydrogenase complex is deficient in cell. What happens? In this case, there is not formation of acetyl CoA. There will be no Krebs cycle. When there is no Krebs cycle, then cell gets energy only by glycolytic pathway. So the energy supply by TCA cycle is blocked. As a result, there is no sufficient energy inside the cell. So patient gets neurological symptoms, as our brain gets energy only. Our brain relies on TCA cycle for its energy supply. So if PDH complex is deficient, there is neurological symptoms. Same happens in case of arsenic poisoning. If there is arsenic poisoning, arsenic inhibits E2 domain of PDH complex. When E2 domain is inhibited, same, there will be no acetyl CoA. TCA cycle is blocked. When TCA cycle is blocked, there is not sufficient energy. As a result, neurological symptoms are produced. So this is all about today's video. Next we will study TCA cycle in the upcoming video, the reaction and regulation at different step of TCA cycle. Thank you.